Hello. So, very good evening to the student who have joined. Can you please let me know if my voice is audible to you? And please let me know if you can see the if you can see my video. Thank you for confirming. Now, please let me know if the PowerPoint presentation is visible to you or not. Is the class PPT visible? Okay, thank you for confirming. We will start at 6 p.m. sharp, so please wait till then. Today it is our week 7 live session. If you have any doubt or any queries from our last live session, which was our class 6, you can let me know now because we have couple of minutes till we will start our session. Okay, thank you so much for your wishes. We will start the session shortly, so please wait till then. So is there any query from last class? If yes, you can let me know now. Or if you have any query from previous any class, you can let me know. We have couple of minutes left till we will start our class. So last class was basically we have discussed about the central dogma of molecular biology. We have discussed about the all the things like related to the transcription, translation, replication, and all these things we have discussed mainly. Today the topics are mostly similar. There are a few new things which I will discuss today, and but all things are related to the central dogma itself. So that is why I am asking if there is any doubt or anything you have not understand from you have not understood from last class or previous any class, you can let me know now. Let us wait for 2 to 3 minutes. Let's see if more students join or not. Then we will start today's session. Okay. Okay, so it's 6 p.m. Let me start with the introductions then. Meanwhile, more, if more students join, they can join automatically. So, very good evening to the students who have joined. My name is Mousumi and I'm a PMRF fellow from the Department of Chemistry and Chemical Biology, IIT ISM Dhanbad. I am your TA for this NPTEL SWAM course of Organic Chemistry in Biology and Drug Development. Today it is our week 7 live session. So, as I have mentioned earlier also that in our previous lecture, we have discussed entirely about the central dogma of molecular biology, that is about the DNA replication. The transcription means the synthesis of RNA from the DNA, the translation, the synthesis of the protein from the RNA, and so on. We have discussed regarding various enzymes that are involved in these processes, and also we have discussed what is the sequence, what is the chronology, and what are the what is the thing, how it happens in general. Okay, so it was our last class, our week 6 live session. So today it is our week 7 live session. Before going into the course content for today's session, let me clarify a few things that there will be this kind of problem solving session every week. These sessions will be on every Friday from 8 to 6 p.m. Okay, these sessions are live on Google Meet platform. This is the link. The link is same which you have used for joining today's class, and this link will remain same for our entire semester. Okay, so you can write it down anywhere, you can copy it anywhere and use it for joining the next class. Otherwise, you are reminded regarding the online classes one day, a day before of the class. Okay, so if the important thing is if any network disruption happens from my side, do not leave the class immediately. I will try to rejoin as soon as possible. Okay, and if network issue happens from your side, the call is always open, so you can rejoin back at any time. I am recording this session. Okay, that is why I have muted your microphones and cameras, but the chat box is always open and I am monitoring the chat box every now and then. So if you have any doubt, any query, or you want to tell me anything, you can let me know through the chat box. Okay, the chat box is always open and I am checking the chat box every now and then. Since I am recording the session so that I can record it seamlessly, I have muted your microphones and cameras. Okay, but the chat box is open and we can use the chat box for communicating. If still any doubt persists after class, you can drop an email to me. Okay, I will answer your queries via email if it is possible, or otherwise if your query is it cannot be answered by email, I will discuss it in the upcoming class. Okay, this presentation and class recording, it will be shared to you. Where you have to find this presentation and class recording, I have already mentioned it couple of times, but I will show you again that you have to go to the SWAM course page. Okay, where you will find there is a left panel for entire weekly updates of every classes. Here you will find that at the end we will find the problem solving session 2025. If you click on the problem solving session recording, you will be redirected to a page where you will find a Excel sheet which contains that particular week's course the PPT and also the that particular week's recording. Okay, you can download the PPTs from that particular Google Drive, which looks like this. You can download it from here, and for the classes you can watch the classes from the YouTube. Okay, because it is uploaded on YouTube. The link will be shared to you. So today it is our week 7 live session, and as I have already mentioned that last class we have discussed about the central dogma. Today we are discussing things which are a little bit different. So today basically we will discuss more about molecular biology. We will discuss different topics of molecular biology, and there will be some relation with our central dogma itself, and then we will discuss a little bit about the chemistry of cofactors and coenzyme. We will discuss what is a cofactor, coenzyme, and how chemistry is related to these biological aspects. Okay, so this is our today's week's course content, that is a week 7 course content. And what we do every week, I have already I think have mentioned in last classes also that I will show you around 20 MCQ questions. Each question will have 4 different options. Okay, so what you have to do is you have to identify A, B, or C, or D, which of the following will be the correct option for a particular question, and you have to let me know in the chat box. Answering during the class does not require, it will not cost any marks, or it will not have any kind of negative marking if your answer is wrong. That is why you can answer freely whatever you think is the correct answer. Let me know it through the chat box. I will show, after checking your answers in the chat box, I will show you the correct answer, and that particular topic we will discuss in detail so that you can easily write in your final examinations also. Okay, And if and also, yeah, so there are some assignments which are posted weekly. That assignments have marks, so you have to be very careful while answering the assignments which are given in the SWAM portal. Okay, so let us start with today's session. If there is any doubt, I have already asked that if there is any doubt, you can let me know. Otherwise, let me start with the first question of today's session. Okay, okay. So the first question of today's session, the problem number one, it says that which of the following statements is true regarding the restriction enzyme? Now, whenever there is a true false kind of question, please be very careful, okay, because many students they make mistake in this kind of question. So read the options very carefully. So which of the following statements is true regarding restriction enzymes? Your options: number A, these are endonucleases; number B, these are exonucleases; number C, this cannot recognize the palindromic sequences; and number D, these digest the single strand DNA at specific sites. So which of the following statements is true regarding restriction enzymes? I will again repeat. These are endonucleases. Number B, these are exonucleases. Number C, this cannot recognize palindromic sequences. And number D, these digest single strand DNA at specific sites. Which of the following statements is true regarding the restriction enzymes? I will wait for some time, then I will show you the correct answer. I will check the chat box and then I will show you the correct answer. Let me wait for a minute or two. Read the options very carefully and then write in the chat box A, B, C, or D, which of the following you think is the correct answer for problem number one. Which of the following statements is true regarding restriction enzymes? Option number A, these are endonucleases. Number B, these are exonucleases. Number C, this cannot recognize palindromic sequences. Number D, these digest single strand DNA at specific sites. Let me check the chat box now. Okay, so I got two different options as answer. One is number A and another is number D. Let me check A or D, which of the following is the correct answer, or is there any different answer which is correct? So the correct answer is number A. These are endonucleases. Okay, so yeah, there was a correct answer. So the statement which is true regarding restriction enzymes is number A, which is endonuclease. Okay, these are endonucleases. So what is restriction enzyme? So restriction endonucleases are a family of enzymes that cut each DNA at specific sequence within the DNA. Alternatively, they are called restriction enzyme. A restriction enzyme can cut the DNA when it recognizes a specific sequence, and these specific sequences are called the restriction sites. So this is very important. The restriction sites, we will discuss about it in detail in our upcoming slides, okay, because this is a very important. So wherever the restriction enzyme cuts the DNA, that also there are specific some sequences of the nucleotide or nucleic bases where this particular restriction enzymes can cut, and it varies with the enzyme. Okay, so that these sites are called the restriction sites. So what are the applications of restriction enzymes? So isolated restriction enzymes are used to manipulate DNA for a different specific application. They are used to assist insertion of genes into plasmid vectors during the gene cloning and protein expression experiments. Restriction enzymes can also be used to distinguish gene alleles by specifically recognizing single base changes in DNA known as single nucleotide polymorphisms. Okay. So, restriction enzymes, also called restriction endonucleases, are proteins produced by bacteria that act as a molecular scissors, recognizing and cleaving the double-stranded DNA at or near specific short nucleotide sequences. They are essential tools in molecular biology for genetic engineering and DNA analysis. How they work? Restriction enzymes are a part of a natural defense mechanism of bacteria called the restriction modification system, which protects against the invading foreign DNA such as bacteriophages or viruses. Recognition: each restriction enzyme recognizes and binds to a specific and unique sequence of DNA, typically 4 to 8 base pairs long, known as the restriction site. These sequences are often palindromic, reading the same forward and backward on the complementary DNA. Cleavage: once bound to the restriction site, the enzyme makes two cuts, one on each strand of the DNA double helix. Protection: to prevent the restriction enzyme from cutting its own DNA, the host bacterium produces corresponding modification enzyme, say methyl transferase. This enzyme adds methyl group to DNA of the same recognition site, protecting it from cleavage. Okay, so yeah, this are all about the restriction endonucleases or restriction enzyme. So number A was the correct answer. Is there any doubt about restriction enzymes? If there is any doubt, you can let me know. Otherwise, I will move forward and I will go to the next question. So I hope there is no problem. So yeah, number A was the correct answer. Okay. Now let me go to the next problem. So problem number 2, it says that for which restriction enzyme requires only which restriction enzyme requires only magnesium 2 ions for restriction. Your options are number A type 1, number B is the type 2, number C is the type 3, and number D is all of the three. Which restriction enzyme requires only magnesium ions for restriction? Type 1, type 2, type 3, or all of these three requires only magnesium ions for restriction? Read the question very carefully and then let me know in the chat box A, B, C, or D which of the following you think is the correct answer for problem number 2. For which restriction enzyme only magnesium 2 ions are required for restriction? Again, I will wait for a minute or two, then I will check the chat box. Which class of restriction enzyme requires only magnesium plus 2 ions for restriction? Type 1, type 2, type 3, or all of these three? Which restriction enzyme requires only magnesium 2 plus for restriction? Let me check the chat box. Let me see if there is any answer. Okay, I got two answer. One is saying it is number B, another is number C. Let me check which of the following is the correct answer for problem number 2. Okay, so problem number 2, the correct answer will be, it is number B, type 2. So type 2 restriction enzyme requires only magnesium 2 plus 2 ions for the restriction. So what are the types of the restriction enzyme? So there are mainly three types of the restriction enzyme. The type 1, let me highlight it. Yeah, it recognizes specific sequence, but then track along the DNA like 1000 to 500 bases before cutting one of the strands, releasing a number of nucleotides around 75 where the cut is made. A second molecule of endonuclease is required to cut the second strand of DNA. For example, EcoK, it requires magnesium plus 2, ATP, and SAM. SAM means S-adenosyl methionine. Okay, so these cofactors are required for its restriction function. Okay, then we have the type 2. Type 2 recognize a specific target sequence in DNA and then break the DNA both strands within or close to the recognition site. For example, for E. coli, and it usually requires only magnesium 2 plus for restriction. Then we have type 3. Type 3 is the intermediate properties between the type 2 and type 1, and they break both the DNA strand at the defined distance from the recognition site. For example, HGAI, and it requires magnesium plus 2 and ATP. So as you can see, the type 1, it requires magnesium plus 2, ATP and SAM. Type 2 only requires magnesium plus 2, and type 3 also requires magnesium plus 2 and ATP. So as their question has asked to identify which restriction enzyme requires only magnesium 2 plus, so your answer will be yes, number 2 is the restriction enzyme which requires only magnesium plus 2. So type 2 restriction enzymes. So restriction enzymes are made up of the single subunit cleave within or at short distance from DNA recognition site. They are simple enzymes having single polypeptide. They have separate methylase and endonuclease activity. It requires mostly magnesium plus 2 as cofactor. This enzyme performs only the restriction function. It is the most common restriction enzyme used in the gene cloning. The recognition and cutting site is the same one. They generally recognize 6 nucleotide. Some recognize 4, 5 or 8 base pair also, and these are the examples. So the nuclear structure for type 1, the structure is multimer and heterotrimer, and for type 2 is it is homodimer. Type 3 is also homodimer. Type 4 is relatively new, and the structures are not that much well studied till now. Okay. Recognition site pattern, two sites in any orientation. For type 1, small 4 to 8 base pair, usually palindromic. Type 3, two sites in head-to-head orientation, non-palindromic, and type 4 has weak specificity. Cleavage site, type 1 variable distance from recognition site, non-specific cleavage. Type 2 cleavage within cleavage within or outside the recognition site, and then we have type 3 cleavage on one strand, leaking activity 24 to 25 base pair from the recognition site. Type 4 methylated only. Okay, cofactors ATP, magnesium ion to SAM is for type 1, magnesium plus 2 for type 2. For type 3, we have ATP, magnesium plus 2 and SAM. For type 4 is ATP and GTP. As I have mentioned, type 4 is not that much well explored till now. So classification, restriction enzymes are categorized into several types based on their structure, location or cleavage and cofactor requirements. Type 2 enzymes are most widely used in molecular biology labs due to their predictable cleavage sites. Type 1 are complex multi-subunit enzymes that cleave at random site far from the recognition sequence. They require ATP, S-adenosyl, N-methionine and magnesium. Type 2 is the most common type. They are simpler enzymes that cut at or very near to their recognition site and usually only require magnesium plus 2 as cofactor. Type 3 multi-subunit enzyme that cut a short specific distance away from the recognition site and require ATP and magnesium. Type 4, they cleave only modified DNA such as methylated DNA. Type 4, they utilize or guide RNA specific sequences for cleavage as seen in the CRISPR or Cas9 systems. Okay. So these are all about the type 2 restriction enzymes. Is there any doubt or is it clear? Let me check. Okay, so I hope it is clear. If you have doubt, you can let me know now, or at the end of the session also you can let me know if you have any doubt. So yeah, number B was the correct answer. Now let us go to the next problem. Problem number 3. It says that consider the following diagram, this one, and identify the correct statement. Here, B is the template strand. Your option number A. Number B, A is the primer. Number C, C is the non-coding strand. And number D, A is the antisense strand. Which of the following statement is the correct one? Number A, B is the template strand. Here you can see here in the figure, this is the strand A, this is the strand B, this is the strand C. Okay, there is a D1, D2 and there is also X. So you have to identify the correct statement. Option number A, B is the template strand. Number B, A is the primer. Number C, C is the non-coding strand. And number D, A is the antisense strand. Which of the following is the correct answer for problem number 3? You have to check this diagram and then let me know A, B, C or D, which of the following you think is the correct answer for problem number 3. I think I have discussed this in our last class when I was discussing about the central dogma. It is very much related to the same things. I have discussed it already. Just you have to correlate the things. Just think what is happening here, what is forming, and then I think you can easily identify the correct answer. I will wait for a minute and then I will check the chat box. Meanwhile, you can let me know A, B, C, or D, which of the following you think is the correct answer for problem number 3. Option number A is the B is the template strand. Option number B is A is the primer. Option number C, it says that the C is the non-coding strand. And option number D, it says A is the anti-sense strand. Which of the following is the correct answer? I will wait for a minute, then I will check the chat box. As I have mentioned, I have discussed it already in our last class, so I think all of you can answer it easily. So let me check the chat box. Let me check if there is any answer. Okay, I don't have any answer for this problem. Okay, let me show you. But I think you have not correlated this from our last class. It is fine. Let me show you what is the correlation between this and our last class's course content. So the correct answer will be number D, A is the anti-sense strand. So why I have said that we have discussed, so we have discussed this thing in the last class, right? About the sense strand, the anti-sense strand, and this transcription process. Let us see. So this is your DNA, right? And in from DNA we have the RNA or the mRNA transcript is can be synthesized from the DNA. You all know, right? So what does this RNA polymerase is doing? It is basically joining these nucleotides and forming a mRNA strand or mRNA transcript. So in our DNA we have two strands. One strand goes for 5 prime to 3 prime to 5 prime from left to right, and another is for from 5 prime to 3 prime from left to right, right? So the strand which goes from 3 prime to 5 prime, it is known as the antisense strand or template strand. Okay, so because it acts as a template for the synthesis of the RNA. Okay, as you can see here, this antisense strand, it is acting as a template, and the mRNA is synthesized by taking this as a template of the antisense strand. Okay, it is taking the complementary base pairs for this antisense strand, and then it is synthesizing the mRNA transcript with the help of the RNA polymerase. Okay, that is why this is the strand which is goes from 3 prime to 5 prime from left to right. It is known as the antisense strand or template strands, and others which goes from the 5 prime to 3 prime, it is known as the sense strand. Okay, so now let us look at the diagram once again. So strand A is the antisense strand. Okay, and then this is the your new mRNA transcript, and so this is the thing. So as in the last class, I think I have discussed about it. I have shown you, and I have also given you a question related to it, like to identify the what is the I mean the coding of what the antisense strand. So this is the same thing. Okay, and this is that's why I have told you that today's class also we will discuss little bit about from our whatever we have discussed in our previous class. So is it clear, or do I need to repeat this particular question? I didn't get any answer in the chat box, that is why I'm asking. I hope it is clear. If not, you can let me know. Okay, if it is clear, then I will go to the next problem. So problem number 4, it says that which of the following restriction enzymes form sticky ends? Your options are number A, Ecori, number B, SMAI, number C, EcoRV, and number D, ALUI. Which of the following restriction enzymes forms the sticky ends? Your options are number A, Ecori, number B, SMAI, number C, EcoRV, and number D, ALUI. Again, I will wait for one or two a minute or two, then I will check the chat box. Which of the following restriction enzymes form the sticky ends? Let me wait for some time, then I will check the chat box to see if there is any answer for problem number 4. Here you have to identify the correct restriction enzyme that forms the sticky ends. Which of the following restriction enzymes forms the sticky ends? Let me check the chat box. Okay, I got two answers. One is number A and another is number D. Let me check which of the following is the correct answer. Okay, so the restriction enzyme that forms the sticky ends will be, it is number A, E. coli. Okay, now let us discuss about it. So first let us discuss what is the sticky end and what is the blunt ends, right? So the way an enzyme cuts the DNA determines the type of the end resulting fragment. Sticky ends, many restriction enzymes make the staggered cuts, leaving one single strand overhangs on the DNA fragments. These sticky ends are very useful in genetic engineering because they can easily specifically reanneal with other DNA fragments that have been cut with the same enzyme. Blunt ends, other enzymes cut straight across the both DNA strand, leaving no overhangs. Fragments with blunt ends can be ligated to any other blunt-ended DNA and but are less efficient in reannealing than sticky ends. So let me show you what is the blunt end and what is the sticky end. For the example, so for example, let us just first I will let me show what is the how it looks. Okay, so in case of the sticky end, as you can see, for example, E. coli, it cuts between adenine and guanine. So as you can see here, the sequence is this. So if E. coli cuts here, so what we will get like this. We will have G here, and we will have A T T C, and in the next strand we will have C T T A A, and we will have a gap, and then we will have G. So here you can see there is a gap in this position in this strand, and there is a gap in this position. So both strand, their gaps are not at the same position, right? But here you can see in the case of the blunt ends, for example ALUI, so you will get in the over, it cuts between the G and C. Okay, so you will see here we will get the end like this. We will have AG, a gap, then we will have CT, and here we will have the like TC, then we will have a gap, then we will have GA. So here you can see the gaps are at the same position, means the ends are blunt. But here the ends are not blunt because the ends, the gaps are not the same position of the two. If the two DNA, if one cut is here and another cut is here, it forms a sticky end. But in case of the two strand, if the cuts are on the same position, then it will form the blunt end like this. Okay, so this is the thing regarding the blunt end, and sticky ends. So sticky ends, the restriction enzyme cuts each of the two strands of DNA double helix at the specific points at the sugar phosphate backbone a little away from the center of the palindromic, but and between the same two bases on the both side. Yes, it will cut at the same two. The same two bases in the both. Strand, but not at the same position. Okay. As a result, a single-stranded overhanging positions are left at the end of the DNA, and these are called the sticky end. The blunt end many restriction enzyme cut both strands of DNA exactly at same nucleotide position, typically at the center of the recognition site. It is called the blunt end. So they sticky ends, they form the hydrogen bonds with the complementary at the cut counterpart parts. The blunt ends, they do not form any hydrogen bonds. The stickiness of the ends facilitates the action of enzyme ligase. In the case of blunt ends, no stickiness, only ligase enzyme helps in joining. For examples are this. So this table has a very good examples like which enzyme is has what kind of ends. So for EcoRV, it has a blunt cutter, and the recognition sequence is like 5 prime GA TA TC 3 prime. Origin of isolation E. coli. For SMAI blunt cutter, it is also the recognition site 5 prime CCC GGG 3 prime. The origin of isolation is it is the Serratia marcinius. Then we have the your Ecori, it is also it is a sticky end producer. BAM-HI, it is also sticky end producer. The recognition sites are respectively is the 5 prime G A TTC 3 prime and 5 prime GGA TCC 3 prime. ALUI is a blunt cutter. ALUI, ACI, PVU, 2, DRAI, ECO, RV, SMAI, all are these are the blind cutters, and these are the respective palindromic system. E. coli, BAM-HI, HIN, III, and XHOI, these are the sticky ends, and these are the palindromic syndrome. You do not need to memorize all the palindromic sequence. There are few very important like BAM-HI, HIND, III, E. coli, EcoRV, so the ALUI. So these are a little bit important. Otherwise, if you can remember, this is fine. And remembering the recognition site is sometimes useful and also important. So if possible, please remember this table. Okay. You do not need to memorize the origin of isolation, only the what kind of cutter it is and what is the recognition sequence. So is it understand? Is it is clear? Is it clear, or do I need to repeat it? If it is not clear, you can let me know. I will repeat it. Otherwise, let me move to the next problem if there is no doubt. Okay. So problem number 5, it says that the palindrome sequence recognized by the restriction enzyme HAI-EE is, your options are number A, AAGG, number B, CTGCAG, number C, GATATC, and number D, GGCC. I have told you that remembering the recognition restriction sites, it is hard, but it is important also that for few restriction enzyme, you have to remember this palindromic sequence which is recognized by it. So problem number 5, the palindrome sequence recognized by the restriction enzyme HAE-III is number A, AAGG, number B, CTGCAG, number C, GATATC, and number D, GGCC. I will wait for a minute, then I will check the chat box. Try to identify what is the correct palindrome sequence recognized by the restriction enzyme HAI-EE. The palindrome sequence recognized by the restriction enzyme HAI-EE is number A, AAGG, number B, CTGCAG, number C, GATATC, and number D, GGCC. Let me check if there is any answer for problem number 5. Okay, I got only one answer, which is saying it is number C. Let me check if number C is correct or not. So, the correct palindromic sequence that is recognized by the enzyme HAI-EE is, it is number D, GGCC. Okay. So HIEEE is a type 2 restriction endonuclease that recognizes a specific 4 base pair DNA sequence, which is 5' GGCC 3' and cleaves it to produce blunt-ended DNA fragment. It was originally isolated from the bacterium Haemophilus aegyptus. The key characteristic, the source is this Haemophilus aegyptus, which is you do not need to remember. Recognition sequence is very important, which you have to memorize. That it is 5' GGCC 3'. Cleavage pattern HAIII cuts directly between the G and C bases on the both DNA strands, resulting in the fragments with no single strands overhand. This type of cut creates a blunt ends. Palindromic sequence, the recognition side is palindromic, meaning it reads the same in 5' to 3' direction on both top to bottom strand. Okay, and isochimesomes, other restriction enzymes such as the BSU, PLI recognize and cut the same GGCC sequence and are therefore considered isomers of the HAI-EE. Biological role in its native bacterial host, HAIII functions as the defense mechanism against foreign DNA, such as the form of the bacteriophages. The bacterium protects its own DNA from cleavage by adding methyl groups to GGCC recognition sites. Applications in molecular biology, the ability of HAIIE to produce the blunt ends make it valuable tool for specific molecular biological applications. For example, cloning. Blunt end ligation is a method used to join DNA fragments while less efficient than sticky end ligation. It is useful when inserting a DNA fragment into the vector without relying on a complementary overhang sequence. Restriction fragment length polymorphism or RFLP analysis. HAIEII was historically used in forensic science and diagnostics for RFLP analysis. Its ability to produce the relatively small DNA fragments and its insensitivity to methylation in mammals made it suitable choice for analyzing the forensic samples. DNA mapping and genotyping, because HAIEIE has a short 4-base pair recognition sequence, its cleavage sites occur more frequently throughout a genome than those of the enzymes that recognize longer sequences. This property can be useful for creating high resolution restriction maps and for genotyping studies. Chromosome folding studies, HAIEII has been used in techniques like chromosome conformation capture, which is also known as 3C, and its variants to study the three-dimensional folding of the chromosomes. Okay. So these are the applications of HAIII restriction enzyme in the molecular biology. So is it clear, or do I need to repeat anything? Okay. Now let me go to the next problem. I'm saying again and again, if you have any query or any doubt, please let me know in the chat box. Problem number 6, it says which of the following statements is false regarding the PCR primers. Your options are: number A, DNA polymerase requires primers to initiate replication. Number B, PCR primers are short and single-stranded. Number C, primers are sequence specific and bind to the particular genome sequence. Number D, higher the length of the primer, lesser will be the selectivity. Which of the following statements is false regarding the PCR primers? Again, I will repeat. Number A, DNA polymerase requires primers to initiate replication. Number B, PCR primers are short, single-stranded. Number C, primers are sequence-specific, bind to a particular genome sequence. Number D, higher the length of the primers, lesser will be the selectivity. Which of the following statements is false regarding the PCR primers? Again, I will wait for a minute or two, then I will show you the answer and I will discuss it. Read the options very carefully, okay? Which of the following statements is false regarding the PCR primers? Number A, DNA polymerase requires primers to initiate replication. Number B, PCR primers are short and single-stranded. Number C, primers are sequence-specific, bind to a particular genome sequence. Number D, higher the length of primer, lesser will be the selectivity. Which of the following statements will be false regarding the PCR primers? Let me check the chat box. Okay. So I got one answer which is saying it is number D. Let me check if number D is the correct answer or not. So the statement which is false regarding the PCR primers will be yes, it is number D. Higher the length of the primers, lesser will be the selectivity. This statement is the false. Okay. So the consideration about the PCR primers, the characteristic of the primer specificity, specific for the intended target sequence, avoid the non-specific hybridization, the thoughts on the primer design, the uniqueness, length, base composition, internal stability, stability from stable duplex with the template under PCR conditions. We have two thoughts on the primer design is the melting temperature, annealing temperature, internal structure, compatibility. Primers used as a pair shall work under the same PCR condition, the primer pair matching. General rules for the primer design, the primer and the amplicon length. Primer length determines the specificity and significantly affects its annealing to the template. Too short, low specificity resulting in non-specific application. Too long, decrease in the template binding efficiency at normal annealing temperature due to higher probability of forming secondary structures such as hairpins. Optimal primer length 18 to 24 base pair for general applications, 30 to 35 base pair for multiplex PCR. Optimal applications amplicon size 100 to 1000 base pair for general application. Avoid greater than 3 kb and 50 to 150 base pair for the real time PCR. Avoid greater than 400 base pair. Okay, so the length it determines the specificity. Too short it is not good because low specificity result in non-specific. Too long is also is not good. Okay, decrease the template binding efficiency at normal annealing temperature due to the higher probability of forming secondary structures as hairpin. So the statement number D, which says higher the length, lesser will be the specificity, it is not true because low specificity will is actually related with the if the strands are too short. Okay. So this statement is false. Other statements, these are correct. DNA polymers requires primers to initiate replication. PCR primers are short and single-stranded, and primers are sequence-specific, bind with a particular genome sequence. Okay. Now let me go to the next problem. So is it fine or do I need to repeat anything? Okay. Let me now go to the next problem. So problem number 7, it says identify the correct statement. Your options are number A, tuck polymerase is used in PCR. Number B, any DNA polymerase can be used in PCR technique. Number C, denaturation of DNA is the second step of the PCR. Number D, none of these. Identify the correct statement. Okay, here you have to identify the correct statement. So whenever there is a, I have already told you many times that whenever there is a correct, incorrect, true, false kind of question, read the options very carefully. Here you have to identify the statement which is correct. Okay, now your options are number A, tuck polymerase is used in the PCR. Number B, any DNA polymerase can be used in PCR technique. Number C, denaturation of DNA is the second step of PCR. Now, and number D, none of these. So, which of the following will be the correct statement? Let me know A, B, C or D, which of the following is the correct statement? Identify the correct statement. Let me check the chat box. Let me check if there is any answer. Okay, I don't have any answer for problem number 7 also. Okay, let me show you and then let us discuss. So the statement which is correct in case of the for problem number 7 will be, it is number A, which says that Tuck polymerase is used in PCR technique. This statement is correct and other statements are not. Okay, so now let us discuss what is Tuck polymerase and how the PCR actually happens. Let us discuss different. So the different steps of PCRs are like this. So your step 1 is the denaturing step, which is at 95-degree temperature. So we have this parent DNA which has a double helix. So first is a or denaturation happens, and we have this two strand. One is 5 prime to 3 prime and another is 3 prime to 5 prime, and we also have the DNA primers here. And then we have the second step, which is the annealing step at the 55-degree temperature. Here the Tuck polymerase is used, the DNA polymerase, which is known as the Tuck polymerase. Let me highlight it. Okay, so here the Tuck polymerase is used in the second step. And what does it do? It basically do there are DNA primers here, and there are nucleotides which will so this section of the DNA will be amplified. It will add to it, and then we have this here we will find the Tuck polymerase, and the step 3 is the synthesizing. Okay, so as you can see, the Tuck polymerase, it is synthesizing the new DNA strands here. Okay, why Tuck and then this cycle is repeated for 20 to 40 times. Okay, so now why this Tuck polymerase is important? So what and why this Tuck polymerase is because due to its thermostability, and it is commercially available. Thermostable DNA polymerase used in PCR to synthesize DNA. During PCR, the reactants are heated up to 95 degrees centigrade. Normal DNA polymerase would be denatured by this high temperature. Tuck polymerase is derived from the eubacterium called Thermus aquaticus. This natural habitat of Thermus aquaticus is the hot thermal springs with ambient temperature of 70 to 75 degrees centigrade. As Thermus aquaticus is adapted to live in extreme temperatures, the Tuck polymerase that is isolated from the bacterium is also capable of withstanding higher temperatures. Tuck polymerase can withstand high temperatures at the denaturation step in the PCR. Okay. So this so now let us just check the all the options. So number 8 says Tuck polymerase is used in PCR. It is correct. Any DNA polymerase cannot be used in PCR because it will not withstand with the high temperature. Because we know proteins or are not so they are get denatured in the high temperature. Denaturation of DNA is the second step. No, it is the denaturation is the first step, followed by the step 2, which is the annealing, and step 3 is the synthesizing. And so number D is also not correct answer. So this is the number A, which is the correct answer. Okay. So is it fine, or is there any doubt or anything? I hope it is clear why Tuck polymerase is important and how it works in case of PCR. Okay. Now let me go to the next problem. So problem number 8, it says that identify the incorrect statement among the following. Your options: number A, cofactors are either small organic molecules or inorganic metal ions. Number B, an enzyme without cofactor is called the holoenzyme. Number C, prosthetic groups are coenzymes which binds permanently with the enzymes. Number D, cofactor is non-protein molecule. Which of the following statement is incorrect? Number A, cofactors are either small organic molecules or inorganic metal ions. Number B, an enzyme without cofactor is called holoenzyme. Number C, prosthetic groups are coenzyme which binds permanently with enzymes. And number D, cofactor is non-protein molecule. Which of the following statement is incorrect? Again, I will give you 1 to 2 minutes. Then I will check the chat box. Identify the incorrect statement among the following. Read the options very carefully because all the options, it might seem that it is correct, or most of the students they actually make silly mistakes during this kind of true false or correct incorrect questions. So read the options very carefully. Number A, cofactors are either small organic molecules or inorganic metal ions. Number B, an enzyme without cofactor is a hollow enzyme. Number C, prosthetic groups are coenzymes which binds permanently with the enzymes. And number D, cofactor is a non-protein molecule. Let me check the chat box. Okay, I don't have any answer for problem number 8 also. Okay, let me show you which of the following will be the correct answer and let us discuss it. So, the incorrect statement will be number B. An enzyme without cofactor is called hollow enzyme. This statement is not correct. So, coenzymes and cofactors, a large number of enzymes require an additional non-protein component to carry out its catalytic functions called as cofactors. A coenzyme, so metal ion, is covalently bound to the enzyme protein is called the prosthetic group. Cofactors are of two types, inorganic ions such as iron plus 2, magnesium plus 2, manganese plus 2, zinc plus 2, and a complex organic molecule called coenzymes. Some enzymes require both coenzymes and one or more metal ions for their activity. Coenzymes function as transient carriers of the specific functional groups. So the enzyme without a cofactor, it's called apoenzyme. Okay, a protein portion which is inactive is called apoenzyme. Coenzyme or cofactor, a non-protein portion which acts as an activator, and the total, the enzyme and coenzyme or cofactor complex, it is known as the hollow enzyme or a whole enzyme which is active. That is why statement number B, which says an enzyme without cofactor is called hollow enzyme. No, an enzyme without cofactor is called the apoenzyme, and a complete enzyme with coenzyme, cofactor, and this protein portion or apoenzyme is called the hollow enzyme. So what is now the difference between the coenzyme and cofactor? So coenzyme is a small organic or non-protein molecule that carry chemical groups between the enzymes, and cofactor is a non-protein chemical compound that is tightly or loosely binds with an enzyme and other protein molecules. Coenzyme is a type of cofactor. Coenzymes and prosthetic groups are two types of cofactors. Coenzymes are basically molecules, cofactors are chemical compounds. Coenzymes are basically organic molecules, cofactors are inorganic compounds. Coenzymes loosely bound to enzymes and they assist the biological transformation. Cofactors, some are covalently bound to the enzyme and aid the function of the relative enzyme. The coenzymes serve as carriers to the enzyme. The cofactor increase the rate of the reaction that is catalyzed by the enzyme. Coenzyme can be removed from the enzyme easily. Cofactor can only be removed by denaturing the enzyme. For examples include vitamins, biotin, coenzyme A are the examples of the coenzyme. Examples include metal ions like zinc plus 2, potassium plus or magnesium plus 2. These are the examples of the cofactors. So these were the differences and similarities between the coenzymes and cofactors. Okay. Now, let me go to the next problem. Problem number 9. Which of the following enzymes is involved in the formation of the recombinant DNA? Your options are number A, helicase, number B, DNA polymerase, number C, ligase, and number D, primase. Which of the following enzymes is involved in the formation of recombinant DNA? Helicase, DNA polymerase, ligase, or primase? I will again wait for some time. A, B, C, or D, whatever you think is the correct answer, let me know. Even if your answer is wrong, it does not matter. But please answer. Which of the following enzymes is involved in formation of recombinant DNA, helicase, DNA polymerase, ligase, or primase? Which of the following enzymes is involved in formation of recombinant DNA? Number A, helicase, number B, DNA polymerase, number C, ligase, and number C, primase. Which of the following enzymes is involved in formation of recombinant DNA? Okay, let me check the chat box. I got one answer which says it is number B. Let me check if number B is the correct answer or not. So the enzyme which is involved in the formation of the recombinant DNA will be, it is number C, ligase. Okay. So what is the recombinant DNA and okay, let me highlight this. So what is the recombinant DNA and gene cloning? So recombinant DNA or rDNA is a form of the artificial DNA that is created by combining two or more sequences that would normally occur together through the process of the gene splicing. And recombinant DNA technology is the technology which allows the DNA to be produced via artificial means. The procedure has been used to change the DNA in the living organisms and may have been even more practical uses in future. So this is the representation, like we have a donor DNA and we have a restriction endonuclease which cuts the desired DNA piece from this donor DNA, and we have a plasmid DNA. Again, restriction endonucleases, they cut the plasmid DNA and creates a gap. Ligase, what does this ligase do? Ligase combines these two, the desired DNA piece and the plasmid DNA to form a recombinant DNA. Then protein encoded by the cloned genes, and this recombinant DNA is introduced into the host cell, and the multiplication section of the clones will give us this recombinant cells. Okay, so what are the steps? So first step is the isolation of the plasmid DNA and human DNA. So we have this bacterial cell, we have this lactase gene or lactose breakdown, and this is the restriction site, and this is the AMPR resistant gene, and this is the bacterium plasmid, and there we have the human gene. Then gene of the interest, we this have the sticky ends, as you can see, the human DNA fragments. So these are inserted here, and these are the recombinant DNA strands. So first is the isolation of plasmid DNA and human DNA. Second, cut both the samples with the same restriction enzyme. Number three, mix the DNAs. They join by base pairing. The products are recombinant plasmid and many non-recombinant plasmids. Then introduce the DNA into bacterial cells that have mutation in their own lactate gene. This is important where you have to introduce it. So you have to introduce the DNA into bacterial cell that have a mutation in their own lactate gene. Okay, then we have the plate the bacteria on agar containing ampicillin and X-gel incubate until the colonies grow. The colony carrying the recombinant plasmid with the intact laxate gene, colony carrying the recombinant plasmid with the disrupted laxate gene. Okay, so these are the bacterial clones. So this is how the recombinant DNA or DNA and gene cloning happens. Okay, so is it fine or is there any doubt in problem number 9? Have you understood the steps of the recombinant DNA and recombinant DNA technology? We will discuss about it a little bit more in our upcoming slides. Okay, so if you have any doubt, you can let me know now. I will discuss it at the end of the session also. Okay, so then problem number 10. Problem number 10 says, which of the following is an example of an advanced cloning technique? Your options are number A, PCR cloning, number B, gypsum assembly, number C, getaway cloning, and number D, all of the above. Which of the following is an example of an advanced cloning technique? Number A, PCR cloning, number B, gypsum assembly, number C, getaway cloning, and number D, all of the above. Which of the following is an example of advanced cloning technique? Let me wait for 2 minutes, then I will check the chat box. Which of the following is an example of an advanced cloning technique? PCR cloning, gypsum assembly, getaway cloning, or all of the above? I will wait for another minute, then I will check the chat box. Let me know A, B, C, or D, which of the following will be the correct answer. Which of the following is an example of advanced cloning technique? Okay, let me check if there is any answer for problem number 10. Okay, I got two answer, number D and number C. Let me check which of the following will be the correct answer. Okay, so yeah. So the technique, which is an example of an advanced cloning technique, will be the correct answer is number D. All of the above are the example of the advanced cloning technique. The PCR cloning, gypsum assembly, gateway cloning, all of these three are the examples of the advanced cloning technique. The recombinant DNA cloning method, also known as the molecular or gene cloning, is a core technique in the genetic engineering used to create identical copies of specific gene or DNA segment. The classical approach relies on the restriction enzyme and DNA ligase to cut and paste the DNA fragments from different sources to create a new and hybrid molecules. The steps of the recombinant DNA cloning method. Number one, isolation of DNA. First, the genetic material DNA containing the gene of interest is isolated from the donor organism. The cloning vector, typically a small circular bacterial plasmid, is also prepared. Both the target DNA vector must be purified to remove the other macromolecules like proteins and RNA. Cutting the DNA using specific restriction enzymes. Both the target DNA and vector are cut at the predetermined restriction sites. Many restriction enzymes produce the staggered cuts that result in sticky ends. Short and single strand overhangs that can be base pair with the complementary sequences. This makes the subsequent ligation step more efficient. Ligation, the cut target DNA and the vector DNA vector are mixed with the DNA ligase and enzyme that acts as a molecular glue. The ligase catalyzes the formation of phosphodiester bonds, permanently joining the DNA fragments to form a single circular recombinant DNA molecule. The transformation, the recombinant DNA is then introduced to the host organism, usually a bacterium like E. coli. The process is known as transformation. The host cells are specifically prepared, for example, with a heat shock or electroporation to make their membrane permeable to the foreign DNA. Selection and screening. After transformation, the host cell are grown on a selective medium, typically containing an antibiotic. The cloning vector contains a scalable marker such as an antibiotic resistant gene, which allows only the bacteria that have successfully taken up the plasmid to survive and grow. For the screening, the steps are used to distinguish between the cells carrying the non-combinant plasmid and those with the desired recombinant plasmid. Multiplication. The selected host cells are cultured in the nutrient-rich environment where they multiply rapidly. Since the recombinant plasmid is replicated with the host cell's own DNA, this process generates billions of identical copies of recombinant DNA. Then step number 7. Finally, the recombinant DNA or protein expressed by the cloned gene can be isolated and purified from the host cells for the further analysis and use. So these are the steps for the recombinant DNA cloning method. Okay. So this I have already discussed, that this is the human cell from the gene from the human growth hormone, and then we have the bacterial chromosome. This is the plasmid where this portion is inserted, the gene for the human growth hormone, and then bacterial cell for containing the gene for the human growth hormone. This is the formed. Okay. So modern variations of the method, while the restriction enzyme-based method is foundational, newer, more advanced cloning methods exist. For example, PCR cloning. This method uses PCR to amplify a DNA fragment, which is then directly ligated into a vector without needing restriction enzymes. Ligation independent cloning, or ILIC, it adds specific short sequence to the ends of the DNA insert that match complementary sequences on the vector. This allows the DNA fragments to bind and repaired by the host cell's enzyme after transformation. Gibson assembly. This is seamless cloning method that uses an enzyme mix to assemble multiple overlapping DNA fragments into the vector in a single one-step reaction. Getaway cloning. This technique is based on the site-specific recombination system of the bacteriophage lambda, allowing thus efficient transfer of DNA fragment between the different vectors. Okay. So PCR cloning, gypsum assembly, gateway cloning, all these are examples of the more modern or advanced cloning techniques. And there is also another technique which is called the ligation independent cloning. Okay. Now let me go to the next problem. Problem number 11. It says which coenzyme is required by the enzyme lactate dehydrogenase. Your options are number A NADP, number B NADH, number C NAD plus, and number D FAD plus. Which enzyme is required by the enzyme lactate dehydrogenase? Number A NADP, number B NADH, number C NAD plus, and number D FAD plus. Which coenzyme is required by the enzyme lactate dehydrogenase? Let me know A, B, C, or D. Which of the following you think is the correct answer? Which coenzymes is required by the enzyme lactate dehydrogenase? NADP, NADH, NAD plus and FAD plus. Let me check the chat box. Let me see if there is any answer for problem number 11. Okay, I got one answer. It is saying that it is number B. Let me check if number B is the correct answer or not. So, the coenzyme which is required by the enzyme lactate dehydrogenase, it will be number C, NAD plus. Okay. So, this is the enzyme lactate dehydrogenase, which is the reaction that it catalyzes, is basically conversion of lactic acid to pyruvic acid. This is the structure of lactic acid, as you can see. This is the structure of pyruvic acid. So NAD plus is the cofactor for lactate dehydrogenase, which when lactic acid forms the pyruvic acid, as you can see, there is a transfer of the hydride, and this particular NAD plus, it takes up the hydride and it forms the NADH. Okay. So this NAD plus is the cofactor of the enzyme lactic dehydrogenase, which converts the lactic acid to pyruvic acid. So many enzymes require additional non-protein substance called cofactor for the reaction to take place. Deficiency of cofactors can arise from poor diet, resulting in the loss of coenzyme activity and subsequent disease like scurvy. Cofactors are either metal ions like zinc or small organic molecules called coenzymes. For example, NAD plus pyridoxal phosphate. Most coenzymes are bound by the ionic bonds and other non-covalent bonding interactions, but some are bound to covalently and are called prosthetic groups. Coenzymes are derived from the water-soluble vitamins and act as the body's chemical reagents. For example, lactate dehydrogenase requires the coenzyme nicotinamide adenine dinucleotide or NAD plus in order to catalyze the dehydrogenation of the lactic acid to pyruvic acid. NAD plus is bound to the active site along with the lactic acid, acts as an oxidizing agent. During the reaction, it is converted to the reduced from NADH. Conversely, the NADH can bind to the enzyme to act as a reducing agent when the enzyme catalyzes the reverse reaction. NAD plus and NADPH are phosphorylated along analogs of NAD plus and NADH respectively, and carry out the redox reaction of the same mechanism. NADPH is used almost exclusively for the reductive biosynthesis, while NADH is used primarily in the generation of the ATP. Okay. So this is very important. What is the differences between coenzyme and cofactor? Earlier I have also, I think, discussed this thing. Like coenzymes are small organic protein molecules that carry out chemical reactions between the enzymes and is a type of cofactor. Cofactor is non-protein chemical compound that tightly or loosely binds with the enzyme and other protein molecules. Coenzymes and prosthetic groups are two types of cofactors. Coenzymes are molecules or mostly organic compound which are loosely bound to enzymes. Cofactors are chemical compound, mostly inorganic compounds. Some are covalently bound to the enzyme. Coenzymes assist biological functions, serves as carrier of the enzymes, can be removed from the enzyme easily. And cofactors aid the function of relative enzyme, increase the rate of the reaction which is catalyzed by the enzyme, can only be removed by denaturing the enzyme. Examples of coenzyme include vitamins, biotin, coenzyme A. Examples of cofactor include the metal ions like zinc plus 2, potassium plus and magnesium plus 2. Okay. So I think it is fine. I will go to the next problem. Problem number 12 says the biosynthesis of gamma aminobutyric acid or GABA from glutamic acid involves number A, glutamate carboxylase as the enzyme and biotin as cofactor. Number B, glutamate decarboxylase as enzyme and TPP as cofactor. Number C, glutamate decarboxylase as enzyme and PLP as cofactor. Number D, glutamate carboxylase as enzyme and PLP as cofactor. The biosynthesis of GABA or gamma aminobutyric acid from glutamic acid involves number A, glutamate carboxylase as a as an enzyme and biotin as cofactor. Number B, glutamate decarboxylase as enzyme and TPP as cofactor. Number C, glutamate decarboxylase as enzyme and PLP as cofactor. Number D, glutamate carboxylase as enzyme and PLP as cofactor. Which of the following is the correct answer? Let me wait for a minute. Then I will check the chat box. Meanwhile, let me know A, B, C or D, which of the following you think is the correct answer. The biosynthesis of gamma aminobutyric acid from glutamic acid involve which enzyme and which cofactor? Your options are number A, glutamic carboxylase as enzyme and biotin as cofactor. Number B, glutamate decarboxylase as enzyme and TPP as cofactor. Number C, glutamate decarboxylase as enzyme and PLP as cofactor. Number D, glutamate carboxylase as enzyme and PLP as cofactor. Let me check the chat box. Let me see if there is any answer for problem number 12. Okay, for I have got two answers for problem number 12, number B and number C. I have got two options. Let me check which of the following will be the correct answer. The correct answer will be number C, glutamate decarboxylase as enzyme and PLP as cofactor. Okay. So GABA aminobutyric acid or GABA acts as an inhibitory synapse in the brain by binding to the specific transmembrane receptors in the plasma membrane of the both pre and postsynaptic neuronal processes. It is most highly concentrated in the substantia nigra and the globus pallidus nuclei of the basal ganglia, followed by the hypothalamus and periaquedural gray matter. So this is major inhibitory neurotransmitter in the mammalian central nervous system, present in the nerve terminal released from the electrically stimulated neurons. GABA functions by binding to the specific transmembrane present in the plasma membrane. So these are the transformations. Glutamate is converted to gamma aminobutyric acid, as you can see, by using this enzyme glutamate decarboxylase, which and also it releases the carbon dioxide. PLP here is acts as a cofactor, and there are several other transformations which is not very important, but I have just given this figure for your understanding. You don't need to remember all these things. Okay. So in mammals, gamma aminobutyric acid or GABA is a primarily synthesized from the amino acid glutamate in a single step reaction catalyzed by the enzyme glutamic acid decarboxylase or glutamate decarboxylase. A second less common pathway of gamma synthesis starts from the polyamines like putrescine. Primary synthesis pathway, the biosynthesis of GABA is closely tied with the body's metabolism of glutamate and citric acid cycle. The main process takes place within GABAergic neurons in the central nervous system and involves the following steps. Number A, glutamate production. The precursor molecule glutamate can be derived from the several sources including the amino acid glutamine and alpha-ketoglutarate, an intermediate of the Krebs cycle. Decarboxylation, the enzyme glutamate decarboxylase removes the carboxyl group from the glutamate, converting it into the GABA. The reversible reactions is the rate limiting step of the GABA synthesis. Cofactor requirement, the enzyme GAD requires the pyridoxal phosphate or PLP, which is active form of vitamin B6 as a cofactor of to the function properly. The GABA shunt, the production and breakdown of GABA are linked through the metabolic loop called the GABA shunt, which partially bypasses a portion of the citric acid cycle. Production glutamate is created from the citric acid intermediate alpha-ketoglutarate via enzyme glutamate dehydrogenase. Glutamate is then converted to GABA by GAD. Degradation, GABA is broken down by mitochondrial enzyme GABA transaminase into the succinic semialdehyde. The succinic semialdehyde is then converted to succinate by the succinic semialdehyde dehydrogenase, and succinate is then re-enters in the cyclic acid cycle, continue to energy production. Okay. So yeah, number C was the correct answer. Now let me go to the next problem. Problem number 13, it says that in TCA cycle, the conversion of the malate to oxaloacetate is catalyzed by dash and the cofactor is dash, filling the blank. So here again you have to identify which of the following is the correct set of the enzymes and cofactors for the conversion of malate to oxaloacetate in the TCA cycle. Your options are number A, malate dehydrogenase and NAD+, number B, malate dehydrogenase, dehalogenase and NADP, number C, oxaloacetate synthesis and NADH, and number D, aconitase and NAD+. Your options, again I will repeat, in the TCA cycle, conversion of the malate to oxaloacetate is catalyzed by dash and the cofactor is dash. Okay, fill in the blanks. Your option number A, malate dehydrogenase and NADP, NAD+, number B, malate dehalogenase and NADP, number C, oxaloacetate synthesis and NADH, and number D, aconitase and NAD+. Let me know A, B, C or D which of the following is the correct answer for problem number 13. In TCA cycle, which enzyme catalyzes the conversion of malate to oxaloacetate, and what is the cofactor for this conversion? So what will be the correct answer for problem number 13? Let me check the chat box. Okay, I don't have any answer for this problem number 13. Okay, let me check which of the following will be the correct answer and then let us discuss about it. So the correct answer is number A, malate dehydrogenase and NAD+. Okay, I just forgot to write it. Malate dehydrogenase and NAD+. Okay. So correct answer is number A. The enzyme malate dehydrogenase is responsible for the conversion of malate to oxaloacetate in TCA cycle, and the cofactor for this same reaction is the NAD+. Okay. So now how it happens? So basically this is the structure of S-malate and this is the structure of oxaloacetate. So as you can see, malate to so this is basically the dehydrogenase, means it catalyzes the oxidation of this to this, means it removes the the hydrogens and the hydrates. So NAD plus is also again the cofactor here, which forms NADH and H plus. This is the TCA cycles. Okay, so first it starts from the pyruvate, which forms the acetyl CoA by the pyruvate dehydrogenase and coenzyme A. Then the acetyl CoA is incorporated into the it is involved in the conversion of in the synthesis of citrate. This citrate is converted to isocitrate by aconitase. Isocitrate is converted to oxalosuccinate by the enzyme isocitrate dehydrogenase. Oxalosuccinate converted to alpha ketoglutarate by the isocitrate dehydrogenase. Then alpha ketoglutarate with the help of the cofactor NAD plus and the enzyme alpha ketoglutarate dehydrogenase converts to succinyl CoA, which converts to succinate with the GTP succinyl CoA synthase. Succinate it converts to fumarate by succinate dehydrogenase. Then fumarate is converted to malate by by the fumarase, malate converted to oxaloacetate by malate dehydrogenase and NAD plus, and oxaloacetate it converts to citrate, then the cycle repeats. Okay, and here the enzyme is citrate synthase. So all that these are the enzymes and the cofactors which is involved in the various steps for the TCA cycle. So the correct answer for problem number 13 will be malate dehydrogenase and NAD plus. You can see from here very clearly. Let me highlight it one for you. So here malate is converted to oxaloacetate. Here the enzyme is the malate dehydrogenase and our cofactor is NAD plus. Okay, so is it clear or do I need to repeat anything? Okay, so I hope it is clear. Now let me go to the next problem. So problem number 14, it says then in PBR 322, P stands for number A prokaryotes, number B plasmid, number C penicillin and number D palindrome. In the PBR 322, P stands for what? Prokaryotes, plasmid, penicillin or palindrome. Again, I will wait for a minute or two. Then I will check the chat box and we will discuss about it. In PBR 322, P stands for what? Prokaryotes, plasmid, penicillin or palindrome. Let me know in the chat box A, B, C or D which of the following you think is the correct answer for problem number 14. Let In PBR 322, P stands for what? Prokaryotes, plasmid, penicillin or palindrome. Let me check the chat box. Okay, I got one answer which says it is number B. Let me check if number B is the correct answer or not. So in PBR 322, P stands for, yes, number B, plasmid. Okay, so this is a little mistake is there. Yes. Okay, so yeah, P stands for plasmid. Now let us discuss what is the PBR322 and what is this plasmid. So plasmid is a small DNA molecule within a cell that is physically separated from a chromosomal DNA and can replicate independently. The term was coined by Lederberg and Hayes and shortly discovered by Tatum. They are mostly commonly found in bacteria as small circular double-stranded DNA molecule. However, plasmids are sometimes present in archaea and eukaryotic organisms. Plasmids carry genes that may benefit the survival of organisms, for example, antibiotic resistance. While the chromosomes are big and contain all the essential genetic information for living under the normal condition, plasmids usually are very small and contain only additional genes that may be useful to the organism under the certain situations or particular conditions. Artificial plasmids are widely used as vectors in molecular cloning, serving to drive the replication of the recombinant DNA sequences within the host organisms. PBR322 is a purpose-built transmit vector, which one of the first widely used cloning vector. This is very important. Okay, and this is because this is a purpose-built plasmid vector and was one of the first widely used cloning vector. It has relatively small size of 4,363 base pair, and now this is important because transformation efficiency is inversely proportional to the size. So this is important because the transformation efficiency is inversely proportional to the size. Okay, and the nomenclature, this created 1977 by laboratory by the Herbert Boyer and University of California, San Francisco. Here, P strand indicates the plasmid. BRs in identifier Bolivar and Rodriguez, the two researchers who developed it. 322 distinguish this plasmid from each other like PBR 325, PBR 327 developed in the same laboratory. This is a 20 to the 4,361 base pair in length and has two antibiotic resistant genes. The gene BLA encoding the ampicillin resistant gene protein and gene TETA encoding the tetracycline resistant protein. It contains the origin replication of the PMB1 and the rope gene which encodes the restrictor plasmid copy number. The plasmid has a unique restriction sites for more than 40 restriction enzymes. 11 of this sites lies in the tetrad gene. There are two sites restriction enzyme like HEN3 and lie within the promoter TR gene. There are 6 key restriction sites inside the AMPR gene. So this is the structure. As you can see, PBR322. There is a E. coli and there is a HEN3 restriction enzymes cut here. This is the TAT zone. This is the AMP gene. Okay, so this is isolated from E. coli and these are the other informations which I have already mentioned. Okay, so yeah, PBR322, P stands for the plasmids and these are the importance of this PBR322. Now, I will go to the next problem. Problem number 14, it says which of the following, which of the two reactions are required to biosynthesize the first derivative of vitamin D inside the body? Your options are number A, disrotatory ring opening followed by 1,5 hydrogen shift. Number B, conrotatory ring for closing followed by 1,5 hydrogen shift. Number C, disrotatory ring closing followed by 1,7 hydrogen shift. Number D, conrotatory ring opening followed by 1,7 hydrogen shift. Which of the two reactions are required to biosynthesize first derivative of vitamin D inside the body? Number A, disrotatory ring opening followed by 1,5 hydrogen shift. Number B, conrotatory ring opening, ring closing followed by 1,5 hydrogen shift. Number C, disrotatory ring closing followed by 1,7 hydrogen shift. And number D, conrotatory ring opening followed by 1,7 hydrogen shift. Which of the two reactions are required to biosynthesize the first derivative of vitamin D inside the body? Number A, disrotatory ring opening followed by 1,5 hydrogen shift. Number B, conrotatory ring closing followed by 1,5 hydrogen shift. Number C, disrotatory ring closing followed by 1,7 hydrogen shift. And number D, conrotatory ring opening followed by 1,7 hydrogen shift. Let me wait for a minute, then I will check the chat box and I will discuss it. Which of the two reactions are required to biosynthesize the first derivative of vitamin D inside the body? Let me check the chat box now. Okay, I got one answer which is saying it is number D. Let me check if number D is the correct answer or not. So the correct answer will be Yes, it is number D, conrotatory ring opening followed by 1,7 hydrogen shift. So 6 pi electron electrocyclic ring opening and closure. So we have two conditions. One is thermal and one is photochemical. So this is the 6 pi system. As you can see, there are 6 pi electron. Two are here, two are here, and two are here. So if the thermal condition it happens, so what will happen in case of the thermal 6 pi electronic ring closing? It is the disrotation. Okay, happens. And in case of the photochemical, what happens? It is the conrotation happens. Okay. So then so this is the thing. So how it happens basically. So let me just draw it for you. So here we have this methyl, as you can see here. Okay. And we have this pi electrons like this. So the here you can see this is the structure. Okay, so so the conrotatory means both will be at the same direction. The rotations will be at the same direction. So if the this is the kind of electron flow which happens, and if the ring closure is conrotatory means it will move to the same direction like this. Okay, if this happens to the same direction, so what happens? This methyl group. So as you can see, we will have a six-member ring. Okay, and the six-member ring will have two double bond here. So one will be above the plane and one will be below the plane. As you can see, if this methyl goes above the plane, this methyl will go below the plane. So this is the conrotatory. In case of the disrotation, both happens in the same direction. Means if it happens in this direction, it will happen in this direction. Although sorry, in case of disrotatory, it will happen on the opposite direction, and in the conrotatory, it will happen in the same direction. So in case of disrotatory, what will happen if the both rotations of these bonds happen in the same direction? We will have both the methyl groups at the above the plane of this ring. So that's how this thing happens. Okay, so remember this thing. So in case of the so let me just erase this. So in case of our 6 pi electronic system, and the in photochemical condition with H nu, the ring closing or ring opening is conrotatory, and in case of the thermal condition, it is the disrotatory. Okay. So the and this is the example of the hydrogen shift, how the hydrogen shift happens. Okay. So this is the 7 dehydrocholesterol, and this is the provitamin D3. And first step is the under UV or sunlight is the 6 pi conrotatory ring closing as for in this position which happens. And then sorry, this is the conrotatory ring opening which is happening in this position, followed by a 7 hydride 1,7 hydrogen shift, which gives us this chlorocalciferol vitamin D3, which is the first derivative of vitamin D inside our body. Okay. So remember, it is the conrotatory ring opening followed by 1,7 hydrogen shift which happens in this reaction. Now I will go to the next problem. Problem number 16 says oxidation of the retinol to retinal is catalyzed by which enzyme? Number A, retinol dehydrogenase. Number B, retinal reductase. Number C, retinal isomerase. And number D, retinal deoxygenase. Oxidation of retinol to retinal is catalyzed by what? Number A, retinol dehydrogenase. Number B, retinal reductase. Number C, retinal isomerase. And number D, retinal deoxygenase. Oxidation of retinol to retinal is catalyzed by which enzyme? The oxidation of retinol to retinal is catalyzed by number A, retinol dehydrogenase, number B, retinal reductase, number C, retinal isomerase, and number D, retinal deoxygenase. Let me check the chat box now. Let me see if there is any answer for this problem. Okay, I got one answer which says it's number A. Let me check if number A is the correct answer or not. So the enzyme which catalyzes the conversion of retinol to retinal is number A, retinol dehydrogenase. Very good. So this is all trans retinol ester, which is which converts to the all trans retinol. Okay, then retinol is converted to the all trans retinal by this retinol dehydrogenase. Let me highlight it for you. So all trans retinol is converted to all trans retinal by the enzyme retinol dehydrogenase. So as you can see the difference here, we have a alcohol which is converted to CHO, means it is a oxidation kind of reaction, and in case of oxidation to dehydrogenase, we all know are the redox catalyst which or redox enzymes, and this is the visual cycle reactions. Okay, the retinol is converted to 11 cis retinal for vision, and their all trans retinol is converted to the all trans retinoic acid for the transcription and regulation, and by aldehyde dehydrogenase. And okay, so this is the correct enzyme is the retinol dehydrogenase for the conversion of retinol to retinal. Now, problem number 17, it says that pyridoxal phosphate is a coenzyme of dash. Your options are number A, aminotransferase, number B, glutaminase, number C, glutamine synthase, and number D, glutamate dehydrogenase. Pyridoxal phosphate is a coenzyme of which enzyme? Enzyme? Number A, aminotransferase, number B, glutaminase, number C, glutamine synthase, and number D, glutamate dehydrogenase. Pyridoxal phosphate is a coenzyme of which enzyme? Aminotransferase, glutaminase, glutamine synthase, or glutamate dehydrogenase. Again, I will wait for a minute or two, then I will check the chat box. Pyridoxal phosphate is a coenzyme of which enzyme? Again, I will wait for a minute. Let me know A, B, C, or D, which of the following you think is the correct answer for problem number 17. Let me check the chat box. Okay, I got one answer which says it is number D. Let me check if number D is the correct answer or not. So pyridoxal phosphate is a coenzyme of number A, aminotransferase. Okay. So this is the mechanism of action. Okay, let me just is the result. So this is the structure of PLP or pyridoxal phosphate. So metabolically active form of vitamin B6 is the pyridoxal phosphate, which acts as a coenzyme for numerous enzymes. This is important. Okay. That pyridoxal phosphate is the metabolically active form of vitamin B6, which serves as a coenzyme of numerous enzymes, of most of which is involved in the metabolic of amino acid. Okay, general mechanisms are the decarboxylation, transamination, racemization, elimination, replacement of reactions, beta group in conversions. So this is the pyridoxal phosphate, which is converted to pyridoxamine phosphate. Okay, and this is the substrate amino acid or amino donor, and this is the product oxo acid. This is the product amino acid and substrate oxo acid. So that's how the pyridoxal phosphate is it basically transfers the amino group from a acceptor to donor molecule, and in general it forms the pyridoxine phosphate is the intermediate which is formed. PLP or pyridoxal phosphate is the active form of the vitamin B6, a critical cofactor for vast of the number of enzymes, particularly those involved in the amino acid metabolism. Its versatile chemical structure allows it to stabilize a variety of reaction intermediates for functioning as the electron sink. For active form of vitamin B6, PLP is synthesized from other forms of vitamin B6, the pyridoxine, pyridoxime in the liver. Key chemical features: the PLP molecule has a heteroaromatic pyridine ring, a reactive aldehyde group, a hydroxyl group, a phosphate group. Enzyme binding: within the enzyme's active site, the aldehyde group of PLP forms a reversible covalent bond called ship base with the amino group of the specific lysine residue. This attachment is known as the internal albumin. When the substrate such as an amino acid binds to the enzyme, it displaces the lysine to form an external albumin. Key biological functions: PLP is a cofactor of over 140 known enzyme activities involved in major metabolic pathways. Amino acid metabolism: PLP is essential for the synthesis and breakdown of the amino acids. Key reactions involve the transamination, transfer of amino acid to the amino acid to an alpha keto acid, decarboxylation, removal of carboxyl group from the amino acid to form important amino-based neurotransmitter like serotonin, dopamine, and GABA. Racemization, conversion of L-amino acids to D-amino acid. Other modification, side chain elimination of the substitution reaction, particularly for the amino acids like serine and cysteine. Glycogen metabolism: PLP is a cofactor for the enzyme glycogen phosphorylase, where the phosphate group assists the breakdown of the glycogen into glucose 1 phosphate. Heme synthesis: it is necessary for the initial step of the synthesis of heme, a component of hemoglobin. Lipid and neurotransmitter synthesis: beyond the amino acid conversion, PLP aids in the production of the sphingolipids, fatty acids, and key neurotransmitters. Hormone regulation: PLP can also be modulated in the action of the steroid hormones such as the androgen and estrogens by the interacting with their receptor proteins. Okay, so these are all about the PLP. Now I will go to the next problem, problem number 18, which says which of the following statements is false regarding eukaryotic mRNA. Your options are: number A, they are derived from the larger RNA precursors; number B, they result from the extensive preceding of the primary transcript before serving as a translation components; number C, they usually have poly A tails in the 3' end; number D, they have both introns and exons in their mature form. Which of the following statements is false regarding the eukaryotic mRNA? I will wait for another minute or two. Read the options very carefully. Which of the following statements is false regarding eukaryotic mRNA? Number A, they are derived from the larger RNA precursors. Number B, they result from extensive preceding in their primary transcript before serving as the translation components. Number C, they usually have poly A tails at the 3' end. Number D, they have both introns and exons in the mature form. Which of the following statements is false regarding the eukaryotic mRNA? Number A, they are derived from the larger RNA precursors. Number B, they result from extensive processing of the primary transcript before the serving as a translation component. Number C, they usually have poly A tails at the 3' end. Number D, they have both introns and exons in the mature form. Let me check the chat box because we don't have much time left. Okay, so I don't have any answer in the chat box. Okay, it's fine. Let me show you the correct answer and let us discuss about it. So the correct answer means the statement which is false regarding eukaryotic mRNA will be number D. They have both introns and exons in their mature form. It is not clear because I have discussed in the last class also this thing. RNA polymerase 2 transcribes the long precursor RNAs. The pre-mRNA must splice or process into the mature mRNAs. Gene in the DNA, we have exons and introns. This transcription or pre-mRNA has both introns and exons, but the splicing process creates the mature mRNA which do not have any intron, it has only the exons, right? So what is the difference between the prokaryotic mRNA and eukaryotic mRNA? The prokaryotic mRNA produced by the transcription and operons on the prokaryotes. Eukaryotic mRNA is produced by the transcription of the single genes in the eukaryotes. This synthesis of mRNA occurs in cytoplasm in case of prokaryotic one, and these are the polycistronic. The eukaryotic mRNA synthesis of mRNA occurs inside the nucleus. It is monocistronic. Prokaryotic mRNA does not contain both 5' and 3' tail. Eukaryotic one contains both 5' cap and 3' tail. The prokaryotic, the transcription is coupled with the translation in prokaryotes. The eukaryotic mRNA, the transcription of the eukaryote occurs after the completion of transcription. Prokaryotic mRNA contains a ribosome binding site, which is the Shine-Dalgarno sequence. The 5' cap of the eukaryotic mRNA is responsible from recruiting ribosomes. Lifespan of the prokaryotic mRNA very short. Lifespan of the eukaryotic mRNA relatively stable. Prokaryotic mRNA undergoes a very little post-translational modification, and eukaryotic ones undergoes a major post-transcriptional modifications. Okay. Now we will go to the next problem. Problem number 19. It says reverse transcriptase requires only which of one the following converting single strand RNA to double strand DNA. Your options are number A all 4 NTPs, number B all 4 DNTPs, number C an RNA template, and number D all 4 DDNTPs. Reverse transcriptase requires which one of the following for converting a single strand RNA into double strand DNA? Number A, all four NTPs. Number B, all four DNTPs. Number C, an RNA template, and number D, all four DDNTPs. I will wait for only a minute, then I will check the chat box because we have one more question which we have to discuss after it. Let me know A, B, C, or D. Which of the following is the correct answer for problem number 19? Which of the following will be the correct answer for problem number 19? Let me check the chat box. Okay, don't have any answer. Okay, let me show you what is the correct answer and let us discuss it a little bit. The correct answer will be number B. All four DNTPs are required for the conversion of single strand RNA to double strand DNA. So reverse transcriptase or RTS is an RNA dependent DNA polymerase. Okay, this is the important thing which you have to remember, that reverse transcriptase is a RNA dependent DNA polymerase, and therefore produces a DNA strand from the RNA template. It has no associated exonuclease activity. Reverse transcriptase is the so we have the single strand RNA template, then polymerization, and using the DNTPs will follow the RNA DNA hybrid, and then it will follow the double strand DNA by RNAs. So transcription and reverse transcription, what are the differences of the process? Transcription is the process of copying genetic information stored in DNA. genome into the complementary strand of RNA. Reverse transcription refers to the reverse process of the normal transcription in which the RNA template is copied to form a cDNA molecule in retrovirus. Encoding the DNA genome into RNA happens in transcription. In case of reverse transcription, encoding of RNA genome to cDNA. Transcription occurs in both prokaryotes and eukaryotes. Reverse transcription occurs in retrovirus. Transcription occurs in cytoplasm of prokaryotes and nucleus of eukaryotes. Reverse transcription occurs in the cytoplasm of the host. Transcription, RNA polymerase is involved. Reverse transcription, reverse transcriptase is involved. Transcription, no primer is used by RNA polymerase, and reverse transcription, lysyl tRNA acts as a primer for the reverse transcriptase. In case of transcription, products of the transcription are used in the protein synthesis, and product of the reverse transcription is integrated into the host genome. So these are the differences between the transcription and reverse transcription. Now the last question of today's session: which of the following statement is false regarding the cystic fibrosis? Your options are: number it is a genetic disease, number B PCR can detect the disease, number C non CF individual, the CTFR gene encodes for the protein that is N-ion channel, and number D, in CF individual, mutation in CTFR gene leads to thick mucus secretion in lungs. Since we don't have much time, I will directly show you the answer because I have to discuss it. So the correct answer will be number C. In non-specific individual, CTFR gene codes for protein that is sodium ion channel. Now, so this is false. Okay, so gene mutation in cystic fibrosis, what happens? So this is normal DNA. In case of normal DNA and the CTFR protein develops normally, reaches the cell surface and becomes open channel for the chloride ion. Okay, so this is our healthy lung cell where we have chloride ion inside the cell, and this is the CTFR protein which is encoded normally, and it opens for the door is open for the chloride ion. The chloride ion can pass through freely. The cilia move back and forth, swapping the mucus out of the lungs. What happens in case of the gene mutation? The door jamming mutation include the G551D, the mutation affecting the LoRa and the Ket-Clevins disables the function of the cell surface. Thick and sticky mucus buildup happens. Okay, then the chloride ions can reach up to the channel surface but cannot pass through because it is called the Doorman problem. This is the CF lung cell with the one problem function, and this is with the two problem, the location and function. The mutant CF protein needs to correct a drug to boost it to the surface. It is also needs a Doorman drug to open the channel so that the chloride ion pass through. This is the common delta F508 mutation. The CFTR protein is made, but it is floats around the inside the cell without the ever reaching to the surface. Okay. So this was the last question of today's session. So today what we did, we have discussed about the molecular biology in detail. We have discussed about various coenzyme cofactor. We have discussed about various biological functions and the coenzymes and cofactor that are involved here in the things, and many things which we have discussed have directly connection with what we have discussed in our last class. Okay, so today it was our week 7 live session. I will share my email ID with you. If you have any doubt or any query, feel free to reach me out through this email. This class presentation and this video will be shared to you, and I have already shown you where you have to find it. You have to go to the SWAM portal. Okay, then you have to go to the first the announcement page where you will find there is a problem solving session in the left panel. If you go to this portion, you will find it will redirect it to a page where you will find the class presentation and also the video. So thank you for joining. Thank you for doing the class and answering. I will close the session for today if there is no problem or no doubt. Okay. If you have any doubt or any query, please let me know via email. Okay. Thank you so much, and I will close the session for today. Let's meet on the next Friday's class, which will be our week 8 live session. Please go through the class materials and let me know if you have any doubt from the previous classes also. If you have any doubt, please let me know. Thank you all. I will close the session for today.